Thank you guys so much for the opportunity to present today. Today I'll be presenting a talk titled Data-Driven Sensor Design using a plant hormone receptor scaffold. And I also want to preface this by saying that a lot of this work that I'll be talking about today comes from this recent PNAS paper. And so you'll see this QR code again as well. But if you're interested in reading that and reading more, all that information is there. And so there we go. This slide is probably a little moot for this seminar series, but I always like to introduce the idea that in vivo perception can be built with biosensors. And especially when you're kind of building biosensors with proteins for small molecule ligand interactors, these systems are often built from existing kind of ligand interacting proteins that can be found in nature. And they carry a lot of advantages, and especially the case that they can be present in living organisms. And so small molecule biosensors have a lot of potential use. These are just a couple examples. You can use them for environmental monitoring, induced responses in living systems. You can utilize them in metabolite pathway and transport discovery. And you can use them to study metabolite dynamics in vivo while the organism is still alive, which is really awesome. My own kind of projects, a lot of the times focus especially around this idea of using them for environmental monitoring. And so Pier 1 is the titled kind of plant hormone receptor that we use a lot and that I'll talk about a lot as a scaffold for biosensor design. So Pier 1 is this really cool receptor that comes from plants. It senses a small molecule called abscisic acid or ABA. You can see the structure here. And what happens in plants kind of naturally in its own system is that when a plant has some form of abiotic stress, it will increase production of this abscisic acid. Pier 1 as a receptor wholly binds to abscisic acid, but upon binding to that target ligand, induces kind of this chemically induced dimerization event with partner proteins. Generally, they're called PP2C phosphatases. But in the case of this talk, I'll be talking a lot about this protein called HAB1. And in plants, this kind of dimerization event induces a stress response. And so Pier 1 is this kind of two protein system that relies on this chemically induced dimerization event. And this carries with it a lot of advantages. One advantage is that you can utilize it for this kind of split transcription factor system, similar to kind of a yeast 2 hybrid, where you can have one side of your protein complex fused to a DNA binding domain, and the other side fused to an activation domain, so that when they are in complex, when they're dimerized, you reform a transcription factor and drive expression of a downstream gene. Another example of how these can be useful and a really cool kind of aspect of these kind of protein-based biosensors is that they can be also used in kind of rapid reporting systems like this nanolux system, where you can fuse Pier 1 to one half of a luciferase and HAB1 to the other half of a luciferase. And when they are in complex, they're close enough together to reform that luciferase as well. And that luciferase can emit light. And the advantage of this system is that it's very, very fast relative to waiting for transcription and translation to happen. Pier 1 is also a really interesting system, and CIDs in general are chemically induced dimerizations in general because they carry with them multiple KDs. And so Pier 1 in particular has this kind of micromolar KD with its ligand, but it has this nanomolar KD, really high affinity with its partner protein once it's in complex. And so it means that you have kind of two opportunities to tune here. But additionally, it means, and this is why we think, and I'll talk about this later, Pier 1 is such a kind of good biosensor scaffold, is that the KD doesn't have to be quite so sensitive for the receptor to the ligand. Also, having these two protein systems means that you can tune the different pieces of it to modulate your EC50 and your dynamic range and things like that. And so all the way back in 2015, work was published and done to look into reprogramming the Pier 1 receptor away from kind of its native ligand. And so this is what the pocket looks like, where Pier 1 is binding to ABA. ABA is kind of in this yellow here. You can see the receptor pocket. And so with only 4 substitution mutations, we were able to get Pier 1 to bind not ABA, but this other compound called mamipropamide. And so only, four substitutions made this really nice biosensor, which was really cool. And so much more recently in 2022, work was done to create this double site mutation library, which carried with a really wide breadth of diversity, and I'll talk more about the design of that library later. But I just want to highlight that in 2022, this library was screened against a number of agrochemicals. And after screening, it was able to find sensors for a surprising number of them. And so this kind of tipped us off towards our future questions. And I'll also highlight here, I'll show this type of data quite a lot. This is UC2 hybrid data. And specifically, it's like kind of a stain data of this UC2 hybrid where blue is activation of your kind of split transcription factor system, and then this white coloration is no activation. So you can see a lot of these are sensors as they have this kind of blue activation in the presence of a ligand, and then no activation without. And so from this paper, because it was kind of so successful, one of our questions was kind of what is the scope of Pier 1 binding? What are the different kind of compounds that Pier 1 is able to bind to? Did we get lucky here with this paper, or is it just a really awesome biosensor scaffold? And the next question that I'll touch on in this talk is how can we use kind of data from these general screen libraries to build effective biosensors rapidly and get really nice high sensitivity sensors and find additional ligand interactors. And so to get at this first question on what is the scope of chemical binding for Pier 1, we need to build biosensors. And so building biosensors typically involves kind of having a large amount of protein diversity, having some way to select for against that protein diversity with a chemical of interest present, and then going back and sequencing those sensors. So you need diversity and selection for biosensor development. And so to kind of look at the diversity of Pier 1 pockets that combine to different small molecules, we created what I like to call general screen libraries. There's two in particular that I'll talk about a lot, which are the DSM and TSM library. And so how these were designed and how these were created on the left here, you can see this is Pier 1. In orange is ABA in the middle. And all these green residues, the thing that they all have in common is that they're all within 5 angstroms of ABA, kind of pointing in and being near and around that receptor pocket there. And so in the design of this kind of general screen library, this sequence profile was created. And so how to read this and how to look at this is every column is a different position. So it corresponds to a different residue here that's also highlighted in green. And then all these letters down here are the permitted amino acid substitutions that we allow for or design for in this kind of general screen library. You'll notice that there are some areas where there are big gaps. These were computationally identified with Rosetta as being point mutations that could destabilize the kind of receptor complex. And so these were taken out from that design just so that we don't have to spend more time screening more of these sensors that we're pretty sure won't work. And so recently, we've been using oligo pools and ordering oligo pools we typically order from Twist to really power this rapid library generation. We can make libraries in just a couple of weeks, basically, and then start screening them right away using this kind of method. And the idea of it is to take the sequence profile, that diversity that you're kind of planning in and around the receptor pocket, you block it off into 3 different chunks. And then you can order oligos with all the pairwise substitutions of these different amino acid changes at these different positions within each block. When you get these oligos in a pool, you can PCR amplify them with these kind of barcoded primers and then assemble them back into Pier 1 into a plasmid backbone that we can use in UC2 hybrids. And this really quickly generates our libraries for us. And the 2 libraries that we used a lot to kind of get at this chemical scope question was the DSM-How library, which was a library that allowed for 2 substitution mutations per Pier 1. The reason to constrain it is that as you add more or allow for more substitution mutations per peer one, the amount of possible peer ones that you're screening starts to blow up really, really rapidly. And so this was taken one step further with the TSM library, which is 3 substitution mutations per peer one. And you can see it goes from 35K member library to closer to 500,000 K member library really, really fast. And so as you add 4 substitutions and more, it gets unwieldy. And we're interested also, and we've seen that you don't need so many mutations to get sensors in the micromolar level. And so now that we had our diversity ready to go to kind of get at this question, we needed our selection. The selection is the selection that we've always been using and have been using quite consistently for a long time. We use yeast, and specifically we use yeast-2 hybrid growth-based screens for our sensors, where we transform our libraries into yeast, and then we can plate them in the presence of our chemical on selection media without or uracil. And the selection gene that we're driving expression of is URA3. And so you can pick colonies from these plates and then retest them both with the chemical and without the chemical. And if they grow with the chemical and they don't grow without the chemical, then that is a true sensor in our yeast context. And so to ask how broad of a range Peer 1 is able to bind to these different small molecules, we challenged these 2 general screen libraries against over 2,700 chemicals individually. Predominantly, this library was coming from the Celic L1300 library, which contains a lot of FDA-approved drugs and natural products. But we also added additional compounds, especially in the space of agrochemicals that we were interested in. And so here's some examples of some of these results. We have 1 micromolar, 10 micromolar, and 100 micromolar sensors here. The chemical name is on the left. And this is how the kind of growth-based assays look, where you have no growth on the no ligand plate, and then you start to see growth at different doses. And for this context, excuse me, we tested at 1 micromolar, 10 micromolar, and 100 micromolar for these retests to get data on also like how sensitive some of these sensors and interactors were. And so looking at the whole picture, we were able to get sensors for around 7% of the small molecules that we were able to screen, which we thought was really high and really exciting. And this represented 181 unique small molecules, many of which were FDA-approved drugs, but there was also natural products and agrochemicals and other compounds. And so when you kind of map the relatedness of all these small molecules, you can create this heat map, which is imaged here on the left, where the more red is more distant related small molecules, and the blue is this kind of through line. So these are like the one-to-one of these small molecules. It's just them mapping to themselves, and then everything in between is some amount of relatedness. And you can see that a lot of this heat map is red, right? Which suggests that a lot of these compounds are distantly related. And you can see that also when you look at the structures of these. So we have a lot of things from all the way up in the top left here. We have phenol derivatives. Kind of in the middle here, we have steroids as an example. And structurally, they're fairly different. So this was also very exciting for us. If you are interested in doing screening yourself and wondering about like kind of what kind of targets Peer 1 could be good for, some of the characteristics of these chemicals are represented here. So the molecular weight, it averages around the molecular weight of abscisic acid, or its kind of native ligand, which makes a lot of sense. I'll suggest that most of the time our targets are under 500, this kind of 500 Dalton's line. There's a couple outliers still, but yeah, structurally, I would recommend screening things that are under that kind of line as well. And then this polar surface area in C log P, you can see it has a lower polar surface area and a higher C log P. This suggests that a lot of our hits are better uptaken with passive transport into yeast because we did all this screening in yeast, which also makes sense. And so typically things are going to be transported better as well. And then drug likeness is this compilation of scores, but many of our hits were more drug likeness than the non-hits that we saw as well, which is something that you see often for these kinds of screens. When you dive into the kind of sequences of these different biosensors, you can see that the sequences are fairly distinct for their distinct kind of chemical target and ligand. We were able to identify over 500 unique sensors for the 182 compounds. So many of these compounds had multiple sensors and different sequences for them. And so now that we had this data, we wanted to ask, okay, how can we use this to both improve the sensitivity of the sensors that we got, since many of these were still kind of in the micromolar level of sensitivity, and also how can we use this data to identify additional ligands and make this kind of platform even more broad and applicable to different kinds of small molecules. Effectively, yeah, how can we build effective sensors from this data? And so I'll talk through kind of three examples that we used for this paper on how you can build kind of mature libraries and improved libraries from this data. So this is I'll talk about coumarins, TNT-like structures, and PFAS-like structures as well. So we started with coumarins. Coumarins represented the largest class of natural products that we had sensors for. So it seemed like a reasonable place to start and a place that we thought we could have a lot of success in immediately, at least to improve sensitivity of these sensors. And so we were wondering how we could use the data to inform new libraries. Really simply, what we did is we pulled all the kind of sensors from these different coumarin targets. You can see some of them represented on the left. This is kind of the base coumarin structure that makes a coumarin a coumarin. And you can see the kind of different sequences that you get from these different sensors. And we use this to inform, again, a much smaller sequence profile than the kind of general screen sequence profile that I showed earlier. And we similarly block this off and generate oligos, bulk, and then do bulk assembly with Golden Gate cloning. I'll highlight in the bottom right here, I created a Shiny app that lets you can pull the data from the paper, or you can upload your own sequence profiles yourself to generate FASTA files. So if anybody wants to make their own libraries, I have an app that can help you kind of generate these pairwise substitution combinations yourself and do that really quickly and easily. And so when you assemble this library in particular, you get a library with 77,000 possible pure ones. And importantly, we allowed for more substitutions per pure one than the general screen library, right? So because we subset the sequence profile, we filtered it down, it's much smaller. We could create a reasonably sized library with more substitution mutations per pure one. And in theory, we're using amino acid changes that are relevant to these kind of coumarin-like structures. And so to remind you of what this data looked like with the general screen library, this is our general screen sensor data for Imperatora and Oslo and Gelatin. These are three different coumarins. We were able to get sensors from the general screen libraries at 10 micromolar and 100 micromolar in the case of Gelatin. And when we screened these same compounds against the coumarin library, you can see there's many more mutations than just two. And we were able to get sensors in the low, or the kind of, yeah, definitely low micromolar in the case of Gelatin, and at 100 nanomolar for Imperatorin and Oslo, improving the sensitivity of these sensors, which was really awesome. Even cooler than that, though, in my opinion, is that when we screen coumarin structures that we were not able to get hits for from this general screen library, we were able to get hits for it in this kind of mature library, even at the low micromolar level of sensitivity. And you can see how many mutations it took to get sensors for some of these things. And so this was really awesome and kind of led us to kind of take off with this idea of, okay, can we use this data to get hits for things that we have chemical targets in mind, but we have no data for. And so we started this process with these kind of TNT-like structures. And so we went after TNT because it's a carcinogen. Monitoring TNT can be costly and can require kind of repeat testing. And additionally, TNT would be an interesting test case because it's not a natural product. So it's a little bit different from the kind of things that we screened in this library. And additionally, sensors for it could be useful for environmental monitoring. And so really simply, I used a package called ChemMine R. They have, if you want to be doing this kind of analysis yourself, they have a really nice GUI as well on chemminetools.ucr.edu, where I took the SMILES string for TNT and I just threw it in with all of the hits that we have. That's in the supplemental of the paper as well, and clustered them together with Jarvis-Patrick clustering and also hierarchical clustering. And then I curated these four compounds that structurally were very, very similar, or more, at least out of all of the hits that we did have, was the most similar to this kind of TNT structure. Similarly, we generated this smaller sequence profile from the related hits and allowed for 1 to 9 substitutions per peer one. This created a much larger library. It's a little over 500,000 potential peer ones. And when we used it to screen for TNT, we were able to get low nanomolar hits, once again, for targets that weren't at all in our kind of general screen data. So that was, again, super exciting for us. And so the last kind of class of compounds that I'll talk about that we tested our workflow with were these PFAS compounds. And these are very, very unique structure, totally different from everything that we had in here. And so we wanted to test kind of the whole pipeline with this class of compounds. And so PFAS are polyfluoroalkyl substances. They have, yeah, this fluorinated carbon chain, super cool, really bad for the environment. They're toxic in both soil and water, and they exist in many of the products that people produce today. Additionally, they break down very, very slowly in the environment, and they can bioaccumulate into people. And in people, they cause all sorts of problems from immunotoxicity to liver damage and a lot of things in between. Of particular interest were also this EPA6. These are compounds that either historically have been or are currently kind of regulated by the EPA. And there's a lot of different kind of structures within this class of compounds as well. And so again, yeah, this was a space that we could test the whole workflow for identifying Pure One sensors, from screening with the general screen library to creating those mature libraries. And so 103 PFAS class compounds were screened against the DSM library. From that screening, 86 sensors for 18 of those 103 PFAS compounds were identified. And then subsequently, they were used to generate this subset sequence profile. And from this library, mature sensors were identified. This library expanded the scope to 25 targets. So again, we were able to find new ligands and improved existing sensitivities for many of the sensors that we did have. And a really, really awesome sensor that I'll highlight is this Pure One PFAS, which was screened on PFOS, which is one of the APA6, and responded to it at really low levels, kind of 500 nanomolar EC50, which we thought was fairly good. And when this sensor was screened against the rest of the EPA6, it was able to sense at low micromolar levels, 4 of them, which was really awesome as well. And so hopefully I showed that you can kind of use this data and this platform and this workflow to, one, improve existing sensors that we may have from that general screen data, to recluster in new targets that you might be interested in if you don't have access or don't want to do all the initial screening yourself. Or if you have a class of compounds that you're really interested in, you can do the full pipeline kind of with the general screen libraries, get your data yourself, and then make mature libraries with that as well. And there's countless future kind of small molecule targets that you could go after. Yeah, definitely a lot of possibilities on things that you can go for. And so just to pivot really quickly, I'll talk a little bit more on my current work. Since everything in the past was from that PNAS paper, I'll talk a bit more about things that aren't quite published yet, but I'm working towards. And so currently I'm working to adapt this workflow for an orthogonal Pure One called Pure One Star over here. Especially because, and I'll talk about this in a second, but the Pure One scaffold, a lot of this work is done in yeast. Works very well in yeast, and for reasons, this Pure One Star works better in plants. And so I'll also remind you that a lot of my focus is on environmental monitoring. And one thing I'll mention is that I think that plants can act as a really cool kind of case for environmental monitoring and persistent monitoring over wide areas, whether or not that's like kind of in a field of crops that you're growing in, or even in like kind of Superfund sites where you might have this really wide expanse of area that you need to monitor for a small molecule that can be hard to sense. And then I'll also remind you that the Pier 1 scaffold is a hormone receptor in plants, and specifically activates the kind of stress response pathway. It's especially associated with the drought stress response. And so having this ABA bind to Pier 1 induces that dimerization and leads to that pathway activating. So what that means for us and the Pier 1 sensors that we develop is that if we were to put them back into plants, they will similarly interact with and activate these pathways in plants because they still have their own HAB1s and their own Pier 1s, and these things can kind of still interact with each other. And so this was actually kind of the point of the 2015 paper was this chemically induced drought response with this Pier 1 MANDI sensor. And so when you apply MANDI, you can see that the stomata, or the temperature of the leaves, actually heats up. This is because stomata closes, which is a drought response here. And this happens with the Pier 1 MANDI sensor in the plant here. And so to solve this and to get around this, orthogonal sensor was created called Pier 1 star, and an orthogonal partner protein was created called HAB1 star. And they carry with them two substitution mutations that are sufficient for this orthogonalization to not interfere with Pier 1 or HAB1 and that kind of native pathway. They do this with forming this salt bridge between the D162 on Pier 1 and this R393 on HAB1 star. Another really cool advantage to this is that the Pier 1 star allows for two sensor systems in this Pier 1 context. And so you can see here, this top construct is carrying the Pier 1 star kind of cassette. This bottom construct is carrying just the Pier 1 scaffold. And you can use these sensors in the same cell to report and activate different kind of expression cassettes. But again, for my own interest, Pier 1 star is kind of the ideal sensor scaffold to generate sensors for plants and expand the kind of toolkit and array of different sensors that we have available for plant biology. Because it's orthogonal and because it doesn't activate that drought response, it's more synthetic. And so I once again turn to coumarins because we have so much data for them. And I've been adapting the Pier 1 workflow for Pier 1 star. And to do this, it was, again, fairly simple approach, which was to take all the information for high sensitivity coumarin sensors and embed in that T162D substitution mutation to convert Pier 1 to Pier 1 star. That created the Pier 1 star coumarin library. This library was almost 2 million. I would recommend, if you're making your own libraries, I would keep them under 1 million. This was a very, very large library, and the coverage is, I'll say, a little bit dubious. But again, we get 1 to 9 substitutions per Pier 1 star in this case. And when we screened this in yeast to identify mature sensors for it, I was really happy with it because we were able to get high sensitivity Pier 1 star biosensors at similar levels to kind of the Pier 1 scaffold. So these are at low nanomolar levels of sensitivity, which is really awesome. I've since moved these into our model plant, Arabidopsis thaliana. You can do this really easily by just cloning it into this vector. You can do it even from the colony PCR, from the yeast itself. And the way that this construct works is that, again, it's a split transcription factor system. It works very similarly to the yeast-2 hybrid system where when coumarins are present, you have that protein-protein interaction. And in this case, you drive expression of a set of genes called Ruby. So this is like 3 genes that collectively are called Ruby, induce the formation of this red pigment. And so you can see without chemical, you have this plant that has, chlorophyll, it's green, it looks pretty normal. And then when you drive expression of Ruby, you get this nice red pigmentation color. The Ruby gene is cool in its own right. It comes from beets. So that's where this red pigment is coming from, is kind of that beet root, which is really fun. And when I tested the 3 kind of high sensitivity sensors I was interested in testing, I was happy with this. This is in the T2 generation, and so they're still segregating heterozygote for homozygote. So there's still a dosage thing happening here with these representative plants. But you can see that they're responding well at the low micromolar level in plants. And so we're able to translate these sensors into plants really nicely. And so hopefully altogether through this talk, I've shown how you can kind of use this data-driven sensor design workflow and how it works well for different targets. Um, and I'm currently working to adapt this for an orthogonal scaffold. And currently I'm testing the full Pure One Star pipeline for these really cool set of small molecules that we're interested in for being early indicators of fungal infection in plants to try and get specific and early warning for fungal infection in plants. And so with that, I'd like to thank my funding. Thank you to my lab and the Wielden Lab. And I also want to highlight and thank all the people who worked on this paper that came out of PNAS, including the Whitehead Lab Group and all the names here who are the co-authors on that paper. I also want to give a shout out to another really cool paper on how you can computationally design Pure One libraries as well. This is a paper that on the far right, hopefully you guys can see my mouse, but it came out in Nature Commons or Nature Communications relatively recently as well. So I'll give a shout out to them. And then, yeah, again, if you want to build these libraries yourself, I suggest you check out my Shiny app. It makes generating the FASTA files really easy. And if anybody wants to connect and chat about biosensors or especially also synthetic plant biology, yeah, I'd love to chat and talk about biosensor design and ideas on how we can make them better. So thank you all very much.